Okay, welcome back to the Philippines and hello world. Ayan guys, so much for the negative bacilli. Ayan, so we'll now proceed with your gram positive bacilli. Wait lang, it's not giving. Ayan. So we have your gram positive bacilli. So much with gram negative, manatato. But do know that we still have your miscellaneous gram negative bacteria. So sa finals na to sila, mga miscellaneous. So we'll proceed with the gram positive bacilli. Ayan. So learning objectives, of course, no, in terms of general properties and pathogenesis, no, the different properties and pathogenesis, explain the characteristics, morphological, cultural, and biochemical, and even the serological testing. Pathogenic from non-pathogenic members of the genus, and prepare schematic diagram for identification. Yan. So we've classified no our gram-positive bacilli according to the spore formers and yung mga non-spore forming bacilli. So recall no that your spores is the structure of your bacteria that is resistant no or that gives resistance to extreme temperatures or extreme environments for your organisms. Yan. So we have two genus no that are spore forming. We have your bacillus and your clostridium. So your bacillus no they are large, they are large gram positive rods. Okay, so two species under your bacillus no significant. We have your bacillus anthracis and the bacillus cereus. Okay, so they are endospore forming. So makita ninyo, okay, kani siya is actually your bacillus anthracis. So the spores are centrally located. Yan. So centrally located. Okay, streptobacilli sila most of the time, no? Strep ng sunod-sunod in chains. And then centrally located ang ilahang spores. So the motility varies, no? And mostly they are found in the soil or saprophytic. So they derive nourishment from the dead or decaying matter. Aerobic or facultative anaerobe sila, and bacillus are catalase positive. Ayan, so kung i-catalase mo siya, magbaballs siya. Yan, so spores na siya. So Bacillus anthracis, no, is one of the major pathogen under this genus. So your Bacillus anthracis are large, square-ended, non-encapsulated, or rather encapsulated, but non-motile. So kung differentiate mo siya with Bacillus cereus, your cereus is actually motile, your Bacillus anthracis is non-motile. Okay, so spores are ellipsoidal to oval that are centrally located. Okay? And to subterminally located. However, they are not swollen. Okay? Nalanggid na sila sa gitna. Okay? Occur in long chains or bamboo appearance. So, mura sila boxcar appearance. Okay? Mura sila bamboo appearance because they are in chains. Okay, so we have now the pathogenicity of your Bacillus anthracis, no? So of course, no, with the presence of capsule, no, they inhibit or they resist phagocytosis of our immune system. Wait lang, guys. Ayan. So we have your Bacillus anthracis no produces anthrax toxin. So your anthrax toxin is made up of 3 proteins na exotoxin. So your anthrax toxin has protective antigen, so which binds to the specific cell receptors. This allows entry now allows entry of your lethal factor and your edema factor. So the protective antigen present in the anthrax toxin will allow entry of your lethal toxin and your edema toxin into the cells via membrane channels. Okay? Via or following proteolytic activation. Ayan. So lethal factor now, okay, is the with protective antigen forms actually a lethal toxin, which is the major virulence factor now of your Bacillus anthracis. So from the name itself, no, lethal. Okay, it kills no macrophage, or it kills your cells in general. Edema factor naman, edema factor, um, ano sa tawag ani? Produces or forms your adenylyl cyclase, no? This catalyzes the conversion of your ATP to cyclic AMP, or the CAMP, no? Recall the CAMP, which leads to an increase in the cellular levels of cyclic CAMP, or the cyclic adenosine monophosphates. Wait lang, guys, very sakit na ako pa. Ayan. So, which leads to the increase in the cellular levels of your cyclic ayan. So, this upsets now the homeostasis, or the water balance in your body, which will evade now your immune system. Yan. So, edema factor plus protective antigen forms the edema toxin. So, these three proteins actually disrupts the cell function and eventually will lead to cell death. Okay. So, naman yan. So we have 4 types actually of anthrax, guys. Okay. So anthrax, so we have cutaneous anthrax. So 95% of the cases, okay, kanang associated with cutaneous anthrax. Less severe lang noon siya, however, very very prone to kanang develop we're very prone to developing into malignant or necrotic na pustule. So ayan, transmission of your cutaneous anthrax is through skin abrasions. So kung masamad ka tapos naisa ka person nga naisamad pod og anthrax, no, so skin yan. So or pwede pod na kanan sa soil, di ba, makita ng anthrax. So kung nakay open wound, no, most likely kung naapoy wound, open wound, and then ma-contaminate mo open wound with Bacillus anthracis, it will cause most likely cutaneous anthrax. So, yan, black scar. Yan, this one, kung makita ninyo na yung black. Ayan. So, sa skin, no, 1 to 7 days na siya pruritic papule, and then will be converted into a vesicle, and then it will develop into a necrotic ulcer. So, 7 to 10 days, so the scar will be developed and then dries out, and then it will leave a scar. But 25% of the cases of cutaneous anthrax will lead to sepsis and meningitis, causing death. Okay? Next is pulmonary anthrax. So, medyo 5% lang ang cases na documented ani. However, high ang mortality rate. Okay? High ang mortality rate, meaning mortality and morbidity rate ani. So, 80 to 90% of patients with pulmonary anthrax will die because it is very fatal. So, it is also known as wool-sorter's disease. Yan. So, transmission of that is through the inhalation of spores present in your aerosols. So, if the spores are inhaled, no, the spores inside your cells, or the bacterial cells, will be phagocytosed by the lungs, and via the lymphatic drainage, no, mato sila sa mediastinal lymph nodes, and then will undergo germination. So in germination, di ba, wala na yung spores, di ba, pag dili na extreme ang environment, mag-germinate sila. So mawala tong spore. Pero in germination, that's the time they will produce toxins. Okay, and the development now of sepsis and eventually death. Diba po, guys? Yan. So next is your gastrointestinal anthrax. So from cutaneous, the most common, no, pulmonary anthrax. We also have your gastrointestinal anthrax. Though rare lang siya, but kung makakause mo po siya gastroenteritis, very violent po. Okay? So ingestion of spores or bacilli from contaminated meat, no, or animals. So it causes invasion, ulceration of your gastrointestinal mucosa. And also, the last type of your anthrax is your oropharyngeal anthrax. Can be acquired through ingestion, characterized by edema, mouth and neck pain, lesion on the throat and the and causes also throat pain. And both no can become systemic if not treated immediately, will lead to septic shock and death. So grabe ang effect ni Bacillus anthracis actually once we have acquired it no from the environment. So very ano sila, very fatal, very lethal sila when they infect a human or a host. Yan, so diagnosis of your Bacillus anthracis, no, we can use skin swabs, swabbing the vesicle and the black eschar sa cutaneous anthrax. Okay, so makita nimo, sa imong makitaan, bacilli, gram positive, again, color sa gram positive, purple, with centrally located na mga spores. Okay, sputum, stool, no, pwede natin siya gamiton as lab sa specimen. Blood, CSF, gastric aspirate if magiging systemic na siya. Microscopy, we can do gram staining, India ink, and the fluorescent antibody testing. For culture, we can just use the sheep's blood agar. Okay. Serological testing, we can do gamma bacteriophage, immunohisto, chemical testing, and ELISA or immunohematological agglutination test and your ELISA. So sa India ink no, ang ano yung itsura, okay? Mag-fluoresce siya, okay? Because of the presence of what? Capsule. Na siya halo. Okay, di ba capsulated siya? Isa po siya sa iyahang virulence factor. We have also the fluorescent label antibody testing. So kung makita ninyo, no, directly mag-fluoresce ang bacteria. Okay. The poly-D-glutamyl capsule. So this is the polysaccharide present in the capsule of your Bacillus anthracis. So dito sa BAP, no, ayan, lysis by the lytic phage gamma. So the plaque area, the clear area, the gamma phage was applied. Okay, the plaque results from the phage ability to lyse the bacterial cell. So the lytic phage gamma can destroy your Bacillus anthracis. So we can use or you can apply this test to detect the presence or to to confirm the presence of Bacillus anthracis. So yan, so culture media again, so optimal growth temperature is at 35 degrees. Okay, so we can use just sheep's blood agar, no, so sa BAP kay medium to large colony siya, 4 to 6 millimeters. Gray, opaque, grayish white ang color. Rough texture and cut glass appearance. Okay, and irregular ang yang margin. So that's why ginatawag siya og medusa head. Diba kailan mo ni medusa? Katong naay bitin sa ulo. So kana siya. So lion head. So very irregular ang margin sa imuhang colonies for Bacillus anthracis. So if you can see that on BAP, no, you suspect as Bacillus anthracis kay very and nami dati sa hospital nagka Bacillus anthracis care kay pagtanaw nila sa BAP kay medusa head colonies, so nanggawas din sila kay nahadlok sila because it might be anthrax, pero it turned out na dili di ay. So yun, so beaten egg white ang yang consistency, no, non-hemolytic sila, gamma hemolytic. And we can do or we can plate also them sa special media like PLET, no? PLET is polymyxin lysozyme endothalus acetate, which enhances the capsule of your demonstration of capsule of your Bacillus anthracis. So sa gelatin agar naman, okay? Sa gelatin agar, so mag-exhibit siya inverted fear 3 appearance. Ayan. So yung nga ni siya, inverted fear 3 appearance sa gelatin agar. Your Bacillus anthracis. Ayan. So inverted fear 3 appearance, kani fear 3 appearance. So we can also do the string of pearl test with 10 units of your penicillin disc. So what is this testing? We can test this one, no, to confirm again the presence of Bacillus anthracis. So what will you do is you stick the organism on MH. What is MH? Mueller-Hinton agar. So you use 10 units of penicillin disc and place over the stick with the cover slip applied. Okay? So, incubate nimo siya at 4 to 6 degrees at 37 degrees. So, katong nasa cover slip, di ba kay MH? So, sa Mueller Hinton, nag-streak ka. Pag streak nimo, mura kang nag-susceptibility test ba? Butangan nimo penicillin. Okay. So, katong penicillin karon i-cover pag yun nimo na siya o cover slip. Okay? So, kanang cover slip karon, na nasa plate na no, with the penicillin and the bacteria, incubate nimo. Then after incubation, pagka next day, kuhaon nimo ang cover slip and then you view it under the microscope. So, that is the string of pearl test. So, they will exhibit like this, no? Mura siya pearl, string of pearl appearance. Sorry. Yan. So, important biochemical characteristics of your Bacillus anthracis, no? Again, they are non-motive. Okay? So, they produce acid from glucose, sucrose, and maltose, GMS, glucose, maltose, and sucrose. They fail to produce acid on the following carbohydrates and lecithinase. Most of your bacillus are positive for your lecithinase. So, starch hydrolysis and casein hydrolysis, they are also positive. So chemotherapy and control though, the drug of choice for your anthrax is your ciprofloxacin. Your penicillin G, streptomycin, gentamicin are previously used. However, they develop resistance to those antibiotics. So the ciprofloxacin now is that drug of choice. So control measures of our Bacillus anthracis, no, disposal of animal carcass by burning. So because para ma-destroy ang spores, decontamination by autoclaving because di ba autoclave can destroy spores. Use PPE in handling infected animals and byproducts, and active immunization, especially sa mga at risk, no, of this at risk occupational hazards, no. Ayan, especially sa pulmonary anthrax. So it is important to note, no, that your Bacillus anthracis is a potential agent of bioterrorism. So in 2001, actually, anthrax spores were mailed in envelopes in the US, causing inhalation and cutaneous or pulmonary and cutaneous anthrax. So pwede gyud siya gamiton as bioterrorism agent. The same with Yersinia pestis. Okay, so pwede gyud siya gamiton. Smallpox can also be used mo for bioterrorism. Ayan. So your Bacillus cereus naman, the same with Bacillus anthracis, they can be found in the soil and even water. So Bacillus cereus, no, compared with anthrax, this causes now food poisoning lang. Okay, so food in rice, cereal grains, no, from the name itself, cereus, no, sa cereal grains, vegetable meats, no. And they resist pasteurization. So makalusot sila sa sterilization process katong ultra high temperature, short time na pasteurization process or sterilization process of your dairy products. No, they resist that one because again, the presence of spores. They grow at low temperature. Similar to Bacillus anthracis lang yun siya, but again, motile si cereus, si anthracis non-motile. They are beta hemolytic. Okay, beta hemolytic sila. And small, shiny, compact, or large, feathery, and spreading colonies. So virulence factors of your Bacillus cereus, we have your hemolysins, enterotoxins, and phospholipase enzymes. So two types of foodborne disease caused by Bacillus cereus. We have the short incubation form, which causes vomiting from the production of emetic toxin, okay, produced by your Bacillus cereus. The emetic toxin produced by your Bacillus cereus is heat stable, causing vomiting within 1 to 6 hours after ingestion. So, fast siya. Short lang incubation time niya. 1 to 6 hours. Upon ingestion of Bacillus cereus, it will form emetic toxin. That toxin will induce vomiting. Okay? No need for medical intervention na siya usually, no? Yun. And boiled rice, this is the method, no, kung paano ta maka-acquire of possible acquiring your Bacillus cereus na bacteria. So from boiled rice, no, dili man jud ma-remove ang spores. Okay, di ma-remove ang spores because again, autoclaving lang can destroy spores. So they are not destroyed by cooking. And then left unrefrigerated, so mag-germinate siya. So once mag-germinate again, mag-release of toxin. So in germination, dito pa mag-release of toxins. The longer incubation form naman of your the bloodborne disease caused by your Cereus, after 18 to 16 hours pa siya magawas of signs and symptoms after ingestion. So they release heat, labile, and enterotoxic. So from milk, meat, poultry, and dairy products. It is important to note also know that yet they can cause opportunistic infection, which will lead to septicemia, pneumonia, meningitis, and peritonitis. So sa back, okay, sa sheep's blood agar, they are small, shiny, and spreading colonies and may produce grayish to lavender color. And a characteristic niya is that wide ang hemolytic pattern or beta hemolysis. So, clear gid ang agar guys, basta na ay hemolysis. Kaya lang pag tubo na to, di ba, sa mga strep, kaya walay hemolytic pattern. So, yun. Yan, biochemical characteristics of your Bacillus cereus, they are motile. Okay, they are motile. Hemolysis, wide zone of beta hemolysis. Produces acid from glucose, maltose, and salicine. Okay, fails to produce acid. So, the same actually sila ni Bacillus anthracis. Again, ang kalayaan nila, motile si anthracis. Ay, non-motile si anthracis. Your cereus is motile. Yan. So, this is the difference, no, of your Bacillus cereus and your Bacillus anthracis. Okay, yan. So, treatment of choice, no, for your Bacillus cereus infection, we have your chloramphenicol, aminoglycoside, clindamycin, erythromycin, and vancomycin. So, okay, so let's proceed now with your non-spore forming gram-positive bacilli. So later na nato i-discuss tong another spore forming which is your Clostridium, no? We'll proceed sa mga non-spore forming gram-positive bacilli. So we have here your aerobic and anaerobic. So sa gram-positive bacilli na non-spore forming, meaning they cannot produce spores, they are divided into two. Sa mga aerobic, meaning they can produce or they can survive with the presence of oxygen, and your anaerobic na mga non-spore forming na bacilli. Okay? So yan. So they are classified according to the oxygen requirement. Okay? So let's start with your catalase positive sila na non-branching. So non-spore forming under the rock ay si Corynebacterium and Listeria. So ibalik na to, okay? Ka-aerobic ta first sa kanisang duha na non-branching. Okay? Ayan, ano to? Okay. So Corynebacterium, they are slender, pleomorphic, club shape. Club shape siya kay mura siya club. Ayan. Non-branching na gram-positive bacilli. So they are aerobic or facultative anaerobes. So mao na siyang the first one to isolate this bacteria is si Klebs, isolated in 1883. Si Loefflers naman discovered that it causes diarrhea. So mao yang tawag kay Kleb-Loefflers bacillus. So this Corynebacterium can produce metachromatic granules, okay, called as Babes Ernst granules. Yeah. So your Corynebacterium actually are non-motile, non-spore forming, and catalase and oxidase positive. Three groups of Corynebacterium. So we have your group 1, group 2, and group 3. The human and animal pathogen, the plant pathogens, and the non-pathogenic. Of course, no, unsa lang man atong studyhan diri, ang human and animal pathogen or the group 1. So the most virulent species under Corynebacterium is your Corynebacterium diphtheriae, which causes diphtheria. Okay, mode of the transmission of diphtheria is direct through direct contact. So virulence factor is the diphtheria exotoxin. Recall, no, most of the toxin produced by the gram-positive bacilli are exotoxins because they are gram-positive organism. So exotoxin attacks the mucous membranes of the respiratory tract, causing severe inflammation and formation of pseudomembrane formation in the or diphtheric pseudomembrane formation on your oropharynx. So mura na siya sa inyong throat. Yes, na pseudomembrane formation. Okay? And then the bacteria grows, as the bacteria grows, no more exotoxin is produced, causing more inflammation and eventual necrosis. And if there's necrosis, no, there will be obstruction, no, or respiratory obstruction. So magdito siya ginawa. So yan, she will die. Kani guys, mo niyang diphtheria, kani na siya pseudomembrane formation. Of course, no, kung mag-form sila dira and mas mudaghan, mas more ang production of pseudomembrane, mag-close ang airway, di nakakaginawa. Okay? Ayan. So, diphtheros means leather. Ayan. So, it causes also bull's neck appearance. So, mudako po di mong diri. Dili siya bayok, guys. Diri sa neck. Okay? Pseudomembrane. Ayan. So, diphtheria toxin, it is produced after lysogenic conversion. So, causes epithelial destruction, inflammation. Ayan. So, bacteria plus inflammatory cells plus necrotic tissue forming pseudomembrane will cause suffocation. So, di na siya kaginawa. Okay? So specimen, no, we can use Dacron swabs or the nasopharyngeal NPS. Okay? And pwede po kuag specimen ang pseudomembrane. But usually guys, no, if there's pseudomembrane, dili na gina siya gina. Kaya syempre, no, ang direct contact maya ang mode of transmission. So very careful jud ta when we get samples from pseudomembrane. Pero kung pag-check pa lang kayo pseudomembrane, murag case naman gid siya of diphtheria. So dili na gid siya necessary na culture. Pero if pwede naman, makakuha mag-sample, we can do that one. Microscopy, gram-positive bacilli, non-acid fast, no? Loeffler's methylene blue na agar, no? They exhibit Chinese letters or palisade, okay? And then the metachromatic granules or the Babes Ernst granules is mas prominent ang iyang production of that in the Loeffler's methylene blue. So culture media, no, for your Corynebacterium, we can do the SBAP, the sheep's blood agar, the CNA, colistin nalidic agar, cysteine or potassium tellurite, or the Loeffler's serum media. So this produces narrow beta hemolytic pattern on sheep's blood agar. Sa Loeffler's serum medium, enhances the production of your metachromatic granules. The PTM or the potassium tellurite media, it is a selective media that is used, no, to isolate your Corynebacterium diphtheriae. This produces gray to black colonies. So if the Loeffler's serum media is used, no, it must be subcultured in the modified Tinsdale medium. So black colonies with brown halo will be the identification of the species of your Corynebacterium. Yan. So sa tellurite agar, it produces black colonies. May black colonies siya sa tellurite agar. So these are the following biotypes of your bacteria or Corynebacterium diphtheriae and the differences of the following organisms. Yan, si Hoyle's tellurite agar, selective differential media for Corynebacterium diphtheriae. Yan. So, test, no? Differentiation. Ayan. Generation time. Different ang ilang different generation time. Si gravis, mitis, and intermedius. So, you read that one na lang, guys. So, biochemical reaction, nitrate reduction, na catalase, urease, and pyrazinamidase negatives. Urease and pyrazinamidase negative siya, but catalase positive. Sa his serum sugar, yan, they produce acid from glucose, galactose, maltose, and dextrine. Sa lactose naman, lactose, mannitol, and sucrose, they cannot produce acid. Yan, so virulence test of your diphtheria, no, we can do in vivo and in vitro. In vivo, in animals, but again, wala na tayo animal inoculation test. Subcutaneous tests, we can pwede pod, no? Inject broth from culture into 2 guinea pigs, 0.8 ml. One animal is given 500 units of antitoxin before introduction of the toxin, and the other wala siya antitoxin. So the animal that is not given antitoxin will die because wala man siya antibodies against that toxin. So it will lead to loss of animals. And that's why we cannot do it at the present time. So kung gusto ka mag-try or try, kung gusto ka um, test of virulence test, we can do this one. Yan. So kung in vivo, we can use guinea pigs. So ang isa ka guinea pig daw butangan og antitoxin daan before siya i-inoculatan og diphtheria. Okay? And katong isa walay toxin and inoculate dayon. So of course ang mamatay katong walay antitoxin. Yan. To prove that an isolate can cause diphtheria, one must demonstrate toxin production. So kung in vitro katong animal inoculation, in vivo rather, ang in vitro we can do it in an elec plate. So the organism sa elec plate is strict on a plate containing low iron. Okay? So a filter strip containing the antitoxin antibody is placed perpendicular to the strip. So kung nag-strict kag paana ang imuhang strip no na naay antitoxin kay paana. Okay? And then diffusion of the antibody to the medium and secretion of toxin will occur in the culture media or sa elec plate. And then the zone of equivalence, at the zone of equivalence, a precipitate will form. Ayan. This one. So if there's precipitin line, okay? If there's precipitin line, meaning there is toxin production. Okay? Ayan. There is toxin production. Yan. So kung makita ninyo no, negative control, wala siya precipitin line. Wala ni ana ba? Yan. So, di ba? So, kung kani siya positive control, this is your unknown organism and gibutang nimo tong filter paper impregnated with antitoxin. Okay? So, kung sa kaning unknown nimo, pag strict after 24 hours of incubation, na siya precipitin line, meaning that is positive. So, meaning na ay toxin or na diphtheria toxin. So, Shick test no is a skin test no for your diphtheria. So, what is Shick's test? Shick's test is the injection of toxin intradermally. Okay, then if the patient produces redness or erythematic lesions after in 2 to 4 days, meaning susceptible siya to diphtheria. Okay, susceptible siya to diphtheria. Pero kung if there's no reaction after the inoculation of toxin in the intradermal route, meaning immune na ka. Okay, so meaning nakay present immunity against diphtheria. Okay, remember also no that your diphtheria apan pagtung bata pa ta na naman tay mga tong vaccine nagyatag sa tuwa, one of the vaccine that is given to us is actually the diphtheria toxin. Ayan. O again no, pag walay red, no, walay red, kay meaning immune na ka. Okay, immune ka. If na red, meaning susceptible pa ka to diphtheria. Wala pa ka immunity against diphtheria. Ayan, so toxicity test. So we have the elec plate test, PCR, ELISA. So treatment, antibiotic is not useful in acute infection. So ang imojong ihatag sa patient kay antitoxin na dayo. Okay? Dili na siya antibiotic but antitoxin. Kay mas grabe ang effect sa toxin sa imong diphtheria or Corynebacterium diphtheriae compared with the mere presence of the bacteria lang. Okay? The antitoxin is obtained from a horse serum. Ayan. But if we can use antibiotics, no, penicillin is the most commonly prescribed and oral erythromycin. Okay, ayan. So the prevention of your diphtheria, no, the DPT vaccine below 7 years old, the primary in immunization that is given 3 doses, those doses in at least 4 weeks apart. So kung ka-remember mo sa una, sige tag-injeksyonan, bata pa ta, umaw na ni siya. Okay? So diphtheria, pertussis, and tetanus. Oh, DPT ang tawag na. 3-in-1 siya na vaccine, guys. Diphtheria, pertussis, and your tetanus. Yan. Yeah. So, sa booster dose of your toxoid given at 7 years old and above. Ayan. So, of course, no, prevention of that is the completion of all immunization. Yan. So, kanang 3 doses, kailangan mahumanim na plus your booster. Okay? Treating contacts. Yan. So, if kay direct contact with patients, in patients with diphtheria, no, especially mga healthcare workers, we can give prophylaxis sa ilaha to receive at they should have 500 milligram of erythromycin 4 times a day as prophylaxis against diphtheria. Yan. So, study na mo sa other bacterium, no, or Corynebacterium na sa libro. Okay. So, basahan na lang ninyo other bacterium. Okay, next is we have your Listeria, no? So, non-spore forming yapon ni sila na gram-positive bacilli. So, the most common na organism under Listeria is your Listeria monocytogenes. So, your Listeria monocytogenes are facultative anaerobes. They are short gram-positive bacilli, non-AFP, non-acid fast. Okay. They exhibit tumbling motility, guys. Okay, they exhibit tumbling motility at 22 to 28 degrees Celsius. So, mo na yung characteristic feature. Umbrella motility at semi-solid media. So, mura siya kung si inverted fear tree, si Bacillus anthracis, sa semi-solid media, si Listeria, umbrella motility. Sige lang, pakita natin picture niya. Less or non-motil siya at 35 degrees to 37, pero pag 22 to 28, ma-motil na siya. Okay, they can survive at 4 degrees, no, sa ref, in low pH and high salt concentrations. So they overcome usually the preservation of food, no? That's why they can cause foodborne, they can cause gastroenteritis. Okay, entry of organism again after ingestion of contaminated food. So catalase and esquilin hydrolysis positive sila. Carbohydrate fermentation positive also with acid but no gas production. So source of infection again contaminated food, no? And undercooked food. Yan. So high risk ang kasagaran ani kay pregnant females, fetuses, newborns, elderly and immunosuppressed. Okay? So pathogenesis of your Listeria in the fetus, no, is the meningitis. So, di ba, daghan kay cause of meningitis, di ba? So, kung recall nimo to, si strep agalactiae, strep pneumoniae, Neisseria gonorrhoeae, Listeria, and your Haemophilus influenzae. So, it can also, Listeria can cause, sa tawag ni, meningitis, no, among elderly and your fetus, no, sa mga newborns. So early onset of your listeriosis, no, can occur in utero, meaning napa siya sa sulod sa mama. Then it will cause septicemia, pneumonia, brain abscess, or meningoencephalitis, and may result to stillbirth or mamatay siya sa sulod. Yan. Late onset naman, no, they are acquired from the normal flora of mothers during childbirth. Okay? It occurs 1 to 2 weeks after birth. Early onset meaning nag-cross na sa placenta tong bacteria. Yan. So, what's this? This is listeriosis. We have 2 perinatal human listeriosis. Again, the early onset and the late onset. So, granulomatous infantiseptica. This results of utero infection. So it causes neonatal sepsis, postular lesions, granulomas in multiple organs, even in utero, sa sulod pala. And death before or after delivery. No, yan yung magiging effect ng early onset syndrome of your listeriosis. Sa late onset naman, no, it causes neonatal meningitis, no, up to the 3rd week of life, meningoencephalitis and bacteremia among immunosuppressed individuals. Okay? Serotype 4B is Listeria monocytogenes serotype 4B is the common cause of foodborne outbreaks. Yan. So, virulence factors of your Listeria, we have adhesins, okay, internalin A, which interacts with your E-cadherin in the epithelial cells. So, yan. So, masulod siya sa cell. And then, Listeriolysin O, this lyses phagolysosome membrane, di ba? Pag once there's phagocytosis, uh, phagolysosome fusion will occur sa sulod. Pero because of the listeriolysin O, they will lyse or it will lyse the fusion of the phagolysosome. So bypass niya ang phagocytosis. So ACT-A also no acts as polymerization of the actin filaments. So maka-move sila within the cell. And also they can form siderophores. So cultivation, CSF, no, because it causes meningitis, blood, amniotic fluid, no, sa mga early onset na listeriosis. Microscopy, makakita ka gram-positive rods. Motility, optimum temperature of the motility of your listeria is 25 degrees. So ayan, tumbling motility and siya sa microscopy and the umbrella shape pattern sa semi-solid agar. Tanawan ninyo, mura siyang umbrella, di ba? So yun. Cultivation is best at 25 degrees Celsius. They grow slowly at 4 degrees and poorly at 42. So sa BAP kay narrow zone of beta hemolysis, sa 50K soy agar or nutrient agar, smooth, translucent, and blue-green columns. Culture media shapes BAP, tryptose agar, and Pea agar. McBride agar is used for listeria, nalidixic acid media. Biochemical testing, we have catalase positive, ferments glucose 3 halose plus salicin. Treatments, again, these are the following treatments for your listeria. Yan. So these are the important biochemical reactions of your listeria. Camp positive po sila, guys. Okay. So we'll discuss the camp reaction of your listeria. So your camp, di ba, test is used to detect the presence of Streptococcus agalactiae, di ba? Your group B strep. However, your listeria also can be positive with camp. Okay? So na siya camp factor, it can also produce camp factor, but it uses a different known organism. Di ba si camp test for group B, ang yang gigamit nato na strict for that is your Staphylococcus aureus. Pero si listeria, ang atong ginagamit for the known organism, strict perpendicularly to your unknown organism, listeria, is your Rhodococcus equi. Okay, again, your Rhodococcus equi is the known organism strict no in perpendicular to your unknown listeria organism. Okay, so those are catalase positive, no? Ayan, so mga non-spore forming na organism. So now we have the catalase negative, non-branching, and simple branching na organism. We have your erysipelotrix drosophatae. So your erysipelotrix drosophatae causes erysipeloid, okay? And so pa, ayan. So they are non-spore forming, non-motile, no capsule, microaerophilic, okay, and catalase negative. Okay, so single short chains, now long non-branching filaments. They weakly ferment glucose and lactose. So we can use BHI or the brain heart infusion media for the isolation of this organism. So it causes erysipeloid and the raised purplish lesion with pain, itchiness, swelling, but it has no pus. Okay, no nano in the fingers. So the treatment for this organism is penicillin G. Yeah, so laboratory diagnosis of your erysipelotrix is that we can use aspirate, blood, or biopsy specimens. So gram stain reaction, sometimes they may look gram negative because they decolorize easily. Yan. So dapat tama lang dyan ang timing sa ato ang pag-decolorization. Okay, so cultivation sa BAP, they're alpha hemolytic. So compared with your listeria, no, which is narrow zone of beta hemolysis, beta hemolytic yun siya in general. Your erysipelotrix are alpha hemolytic. So, ayan. Kita niyo yung alpha hemolytic. Yung ano yung itsura sa alpha hemolytic, guys? Mura siya o green color. Yan. Sa gelatin media, okay, test tube brush naman iyang growth. So, kung si listeria umbrella, si erysipelotrix kay test tube brush. So, tanaw ni mo, mura siya test tube brush iyang growth. Okay? Tellulite agar, black colonies, no, the same with your Corynebacteria. Yan, so biochemical testing, again, ang iyang distinct no, kay catalase negative siya. Okay? And it produces The only gram-positive rod that produces H2S in TSI and even in SIN is your erysipelotrix. Ayan. So, H2S positive siya. Okay? Yan. Yan, H2S. The blackening of the agar. Yan. Next, we have also your lactobacillus species, no? Lactobacillus are found in the water, in the sewage, normal flora of animals, and normal flora po siya sa ato, okay? Non-pathogenic with little clinical significance, okay? So, the advantage of having lactobacillus, no, that is that it maintains normal homeostatic environment, especially in females, no, because it produces large amount of lactic acid. But however, your lactic acid or production of acids, no, are not the production of lactic acid, especially in females, no, is protective against pathogenic organisms because pathogenic organisms cannot survive in an acidic environment. Ayan. So they grow best at 3 to 4 degrees. Yan. So, biochemical reactions of your lactobacillus, we have non-motile, catalase neg sila tanong. Compared with your erysipelotrix, again, H2S positive siya. Okay? Catalase neg, pareho lang sila. Okay? Non-motile, si erysipelotrix kay motile. Okay? Motile siya, guys. Okay? Asa to? Motile. Diba? Test tube brush. Yan. Test tube brush growth on SIM. Ay, non-motile pala ang erysipelotrix, sorry. So, it is still non-motile. Okay? Yan. Difference nila is H2S production. Yan. So, next, so we have the branching and partially acid-fast na gram-positive bacilli. We have your nocardia. So, your nocardia is actually gram-positive, which is also catalase and urease positive, and they are partially acid-fast. So, we can stain actually them through acid-fast staining. Okay. Ubiquitous and they are saprophytic. They makita sa soil. Non-fastidious, no? Isolation of that, dali lang siya motubo. Ayan. The hip, irregular, waxy aerial filaments ang imong makitaan sa imong agar. It causes opportunistic infections. Okay, especially those who are immunocompromised. Sa gram staining, ano siya guys? Branching. Okay, branching. Ayan. Branching siya, branching. Ayan. Sa acid fast, modified acid fast sa ilaha. Okay, diseases, nocardiosis. So, nocardia asteroides is an infection mostly associated with HIV patients. Ayan. So, inhalation of the organism is the mode of transmission. It begins as chronic lobar pneumonia, which mimics tuberculosis. Okay? Dissemination may occur if left untreated. That's why it can reach as far as the brain. Yan. So, aside from your nocardia, okay, aside from your nocardiosis, it can also cause actinomycetoma. So, it causes Madura foot. Okay, Madura foot, yan. Dili na siya diabetic feet, guys. So, that's an infection caused by nocardia. So, localized, slow progressing chronic infection, destructive but often painless. So, painless lang ni siya, though tanaw nimo sakit siya, pero painless lang ni siya. It caused by a filamentous branching bacteria o perisinocardia or your Madura foot can also be caused by a fungus. Okay? Sa mycoviro na siya guys. It causes cutaneous mycosis. Okay, pero kung bacteria ang reason, sinocardia na siya guys. Okay, Nocardia brasiliensis or Nocardia brasiliensis. Asteroides rather and Nocardia brasiliensis. So, laboratory diagnosis of your nocardia specimen may be sputum, no, because it causes lobar pneumonia, PAS, CSF, and biopsy. So, sa gram stain rods na ay branching, and they are modified acid fast stain positive sila. Cultural characteristics no sa culture media, they are yellow to orange colonies with aerial hyphae. So mura siya o actually o um mura siya o fungus because it has aerial hyphae, but it is bacteria. Urea is positive siya. Growth in milk enhances the acid fastest. So milk agar is actually used for nocardia. Yan. Common isolates, this one are the following organisms. Yan. So these are your nocardia species. Treatment, kani siya. Lesexty, antibiotics and surgical drainage or resection of the tiil or the resection of the tumors. Yan. Yan. So you have your assignment. Please, please, please. Na mo yung assignment. Describe the following procedures. Okay. String of pearl test, Ascoli, Nagler's reaction, Schick's test, and the cold enrichment technique. Okay. Assignment ni siya. Ipasa ni Sako ah. Okay. Yan. So you read na lang about actinomyces, no? Na anaerobic. Okay. The aerotolerant actinomyces. Okay. Yan. So these are actinomyces. Your actinomyces are non-acid fast compared with nocardia. No, so yung actinomyces kay branching gyapon siya, pero ang difference nila is that your nocardia is acid fast or modified acid fast or weakly acid fast. Your actinomyces naman they are non-acid fast. Yan, no mycelium compared with nocardia. Ayan, branching filaments may be produced. Yan, so these are the following organisms associated with actinomyces. So, it causes actinomycosis. Ayan. So, purative and granulomatous infection, which causes pyogenic lesions with interconnecting sinus tracts that contains sulfur granules. The sulfur granules is associated with actinomyces, a species. Initiated by trauma. Trauma. Yan. So, three forms of actinomycosis. It causes cervicofacial, lumpy jaw, okay, thoracic, and pulmonary infection and abdominal ulcers. So lab diagnosis of your actinomyces, pus, sputum, and tissue culture may be done. And filaments break into short chains sa BHI. And sa colonies niya guys, no, it causes, ang appearance niya kay molar tooth. Mura siya o molar tooth nato. Ayan. Nga na iyang tubo sa agar. Molar tooth. Mura siya ka ng bagang nato guys. So treatment, we have penicillin, erythromycin, and surgical excision of the mga lesions and drainage. So ayan, so we are done now with your non-branching, okay, na mga gram-positive organism, branching and non-branching. So now we'll proceed with the gram-positive na spore-forming na anaerobic. Okay? Ganina si Bacillus, di ba, spore-forming po siya, but it is aerobic. So your Clostridium is actually anaerobic gram-positive spore-forming bacilli. So it is important to note that the difference of the two is that aerobic si Bacillus, anaerobic si Clostridium. Aside from that, catalase positive si Bacillus, catalase negative si Clostridium. But both of them are spore forming. Okay? So, swollen po ilang spores. Compared with your Bacillus, centrally located ilang spores. Your Clostridium naman kay swollen. It's either subterminal, central, or terminal spores. They produce true exotoxins. We have your tetanus plasmin and botulinum toxin. Yeah, spore forming. So we have your tetany, botulinum perfringens, and your difficile. So the clinically significant Clostridia, they form spores anaerobically. Yeah. So again, balik-balik na siya. General characteristics, no, these are found in the soil, water, and dust. So the same with your bacillus, no, they can be found in the soil. Okay, and then what's pa? Some are normal flora of the GI tract and human beings. Okay, for animals and human beings, and they are obligate anaerobe, meaning They should not they cannot survive in the presence of oxygen. All are motile. Okay. All are motile in the with the presence of Peritrichus flagella except for Clostridium perfringens, Clostridium ramosum, and Clostridium inoculum. Okay. All can sporulate in vitro except for perfringens, ramosum, and the Clostridioforme. And all have swollen sporangia or non-encapsulated except for these two. Then all are Nagler negative except for the following. Okay. Ayan. So let's start with Clostridium tetani. So we'll just discuss no the significant na mga Clostridium species. So first is we have your Clostridium tetani. So your Clostridium tetani is also known as tuckhead bacillus. Nganong tuckhead man siya, ma'am? Mura siya thumbtacks, guys. Kay ngano, ang young spores kay nasa terminal end. Yan. A drumstick or tennis racket appearance. So tetanus, it causes tetanus. So this one is the one causing tetanus. Entry of organism inside the body. So okay, it will um uh cause necrotic cells, provides anaerobic environment, okay, enabling the organism to germinate. Again, if there's germination, there's release of toxin, which is the tetanus plasmin. Okay, and is produced, released, and absorbed in our body. So virulence factors, so we have two virulence factors of your Clostridium tetani. We have your tetanolysin and tetanus plasmin. So guys, pagaw na kay ko, kumano lang dyan natin eh. Tetanolysin and tetanus plasmin. Tetanus plasmin is the neurotoxin, no? The toxin that goes to the CNS and fix the receptor in the brain and spinal cord. This inhibits now blocks inhibitory neurotransmitters. So they block the inhibitory neurotransmitter. So they will lead to spasms. So 'di ba pag naay neurotransmission, of course no, there will be contraction. Pero dapat manggod napud tay inhibitory para dili pud ta magsigi og contract. Okay? So karon si tetanus plasmin ang iyang action kay inhibit niya ang tetanus ang inhibitory sa neurotransmitter. So kung walay mag-inhibit sa contraction, magsigi ka og contract. That will lead to muscle spasm or it will lead to spastic paralysis. Ayan. So, this one, okay, is the difference of your botulinum and your tetanus toxin. So, your tetanus toxin, this one, okay, si tetani mantano. These are neurotoxin. This is the nerve, diba? This is the muscle. Okay, so this tetanus toxin, no, will inhibit the or will block the glycine release. So glycine release induces muscle relaxation. Okay, so normally, your neurotransmitters will release glycine para mag-relax ang atong muscles. Okay, so your tetanus toxin or tetanus plasmin will block now the release of glycine. So walay relaxation. Dili ta magsigilag contract. Okay, that's why it causes spastic or twitching paralysis or lockjaw. Sige contract. Dili siya mag-relax. Okay, that's the action of your tetanus toxin. Okay? Kung yan na si botulinum toxin, kay lahi po na siya na story. Okay, si tetanus toxin, yung mga to siya, magsige kag contract. Okay, walay relaxation. Yan, so it causes tetanus, no? The clinical manifestation, trismus or lockjaw, rhizus sardonicus, sardonic green, and opistotonos, the arching of the back. So, kani siya, guys. Tadan! Ayan. So, magsigira ka. Yan. So, that's the paralysis caused by your tetanus, Clostridium tetani. Yan. More siya gisapian. Okay? Oh, nga na siya, guys. Sardonic green, ayan. This is the sardonic green. Kanang, ana na siya. Mura siya na stroke, pero wala siya na stroke. It is caused by tetanus or the tetanus plasmin. Ayan, so kaluoy sa mga babies. Ayan, so general characteristic or cultural characteristics of your Clostridium tetani, again, they are gram-positive bacilli with round or terminal spores. Sa blood agar plate, they can produce swarming, okay, with delicate flat edge of projecting filaments. Kung filamentous ang iyahang colonies. And faint lang ang beta hemolysis niya. Yan. For other tests, no, biochem test, we have your positive siya sa gelatinase and indole, but negative siya sa lipase and lecithinase. They are motile, okay, and does not ferment most of the carbohydrates because again, they are anaerobic or anaerobes. Not usually cultured na gitni ang tetanus guys kay based on clinical manifestations pa lang, maka-diagnose na ang doctors. Okay, clinical. Diagnosis is clinical picture and history of injury. If there's injury, no, sa tong patient. So, treatment actually, if left untreated, 50% of that will die or patients with tetanus. But ang treatment niya, dili antibiotics, but against the toxin production. Okay, again, dili siya against the bacteria, but against the toxins produced by your Clostridium. Okay, so pwede ta maghatag og antitoxin, no, 250 to 500 units of tetanus immunoglobulin that binds to the free toxin produced by your Clostridium tetani. Pwede po supportive therapy. We have barbiturates, diazepam. It assist in the breathing. Kaya sige ba contract wala relaxation. So we can do dilators, bronchodilators, which controls the spasm. Okay, ayan. Debridement, debridement of your wound and removal of the necrotic tissue. We can do also antibiotic therapy, though di na siya indicated. But it prevents the growth of Clostridium tetani. So, pwede siya both, no, antimicrobial agents or antibiotics plus your tetanus IG or the tetanus immunoglobulin. So, active immunization. Di ba, gingon na ako sa ganina si DTP or DPT, no? Si diphtheria, tetanus, and pertussis. Okay, so si tetanus, no, active immunization with toxoids. Meaning active immunization. Tama gud tayo tawag na active and passive, guys. Di ba? Amot na ako na-discuss na ko ni prelims. But active immunization, meaning you are exposed to an antigen and you produce immunoglobulins against that antigen. So that's active. Pero kung ang ihatag nimo sa ako kay antibody na diretso, that's passive. Okay? And dili siya long-lasting ang effect pag passive immunization ka. Kung active siya, kaya nag-introduce man kag antigen, so ang imong body nag-create of memory against that antigen, so every time maka-encounter siya ato, nana siya ma-produce na antibodies. So that's active immunization. So kani siya, tetanus toxoids are given. Ay, na-discuss naman ako ng toxoids before, di ba? So, yan. And tetanus and diphtheria vaccine given to adolescents as booster lang. And proper care of wounds contaminated with soil. So, kung masamad ka sa soil with Clostridium tetani, of course, limpyuhan lang dyan siya taro. Okay? Ayan. So, after the Clostridium tetani, we have your Clostridium botulinum. So, your Clostridium botulinum causes botulism, food, wound, and infant botulism. It causes yan. Botulism toxin is a powerful neurotoxin. Okay, it has 7 types, no? A to G, A, B, C, D, E, F, G. Yan. So, 7 types of your botulism toxin, but the most common, si A, si B, and si E. So, botulinum toxin A, B, and letter E. So, signs and symptoms of patients acquiring this bacteria will occur or will be evident, no, 12 to 72 hours of incubation. Yan, so it causes nausea, vomiting, and constipation plus neurological symptoms. Yan. O, kita ninyo, kani siya tuckhead. Di siya tuckhead. Na subterminal lang po ang iyahang spores. Okay? So, motile siya with Peritricus flagella. Produces heat-resistant spores when grown on alkaline glucose gelatin, and heat resistance is diminished at acid pH or high salt concentrations. Yan. So, Clostridium botulinum with subterminal, terminal ends. Ayan. So, mura siya ay ganyan. Yung spores niya kay swollen. Yan. So, virulence factors again, exotoxin A to G, A, B, C, D, E, F, G. The most potent toxin, yan, exotoxins. A, B, and E are common that causes human illness. So, this exotoxin produced by your botulinum or the botulism toxin caused by Clostridium botulinum blocks now the release of acetylcholine in the neuromuscular junction causing flaccid paralysis. So, baliktad siya og effect kay tetanus toxin. So, let's see. The exotoxin also produced by your botulinum toxin is destroyed or easily destroyed at 100 degrees Celsius for 20 minutes. So we can just do boiling, no, para dili ta magkaroon og botulinum toxin. Ayan, so balik ta. So ganina, di ba, si tetanus toxin, it blocks the release of glycine. The glycine, di ba, ang naga-relaxation. Okay, production of glycine induces relaxation sa atong muscles. So kung ma-inhibit si glycine, walay relaxation, magsige contraction. Okay? Baliktad naman si botulinum toxin. Si botulinum toxin naman, it inhibits the release of your acetylcholine. Si acetylcholine releases normally it is released and induces muscle contraction. Okay? So kung walay contraction, sige lang po kag-relax, lupay-pay ka, kumbaga. So that is the effect of the botulinum toxin in our body. It causes flaccid paralysis. Okay? Yan. So wound botulism is rare, no? In germination, of course, it will release the botulinum toxin. So usually associated siya with patients na currently or usually using or constantly using intravenous na method for drug administration. Kung contaminated ang needle, balik-balik gamit ang needle, no? Tapos na eye spores, that's the introduction of your wound botulism. Okay? And infant botulism, growth of organism in the GI tract of infants in less than 1 year. Okay? So ingestion of spores, no? Contaminated siya, spoiled dust and honey, germinates in the GI tract. Okay? And then the botulinum toxin will be produced. So subclinical state to start sudden death, no? It will cause death, no, among infants. So it causes constipation, difficulty in swallowing, muscle weakness, loss of head control. Okay, di ba kay wala naman siya contraction. So sige lang relax. Okay, it causes actually fluffy baby syndrome or it causes crib death. Ayan o, fluffy baby syndrome. Ayan, sabi ko sa inyo. Fluffy baby, relax, puro lang relax guys. Ang na-paralyze na siya, like relaxation. So this is the fluffy baby syndrome. No? Clostridium botulinum spores ingested from baby's food. Ayan, so present with weak sucking response, loss of tone, and circulatory complications. Ayan. So, classic botulinum toxin, foodborne, infant botulism, and wound botulism. Ayan. So, mostly guys, associated siya sa mga canned goods. Ayan. So, muna dapat, kung canned goods ka, dapat stored properly. Okay? And cooked properly. Ayan. So, cultural characteristics of your Clostridium botulinum sa back, they are strict anaerobic. All strains except for type G na Clostridium are beta hemolytic. Si type G lang ang non-hemolytic. Identification of the toxin sa public health laboratory na siya. Ayan, so beta hemolytic. Si beta hemolytic siya. So biochemical testing, wait lang guys ha. Lipase positive, lecithinase negative, indole negative, ferments glucose but not lactose or xylose. Motile and they have oval subterminal spores. treatment, no, mortality rate is 5 to 10% lang. So, just administer trivalent antitoxin. Okay? Um, pero guys, dapat paspas, no, pag once suspected ka with botulinum or Clostridium botulinum, dapat maka-administer dayon ka og um, trivalent na antitoxin, because if not, pag moabot na siya sa nerve endings, no, the antitoxin will no longer be effective. So dapat ko prompt gyud ang imong pag-administer sa treatment. Okay? Next is your Clostridium perfringens. Ayan. So this is also known as the gas gangrene bacillus. Ayan. Encapsulated and non-motile and no spores. Okay? Wala siya spores. Compared with your Bacillus botulinum and Clostridium tetani and Clostridium botulinum na sila spores. Si Clostridium perfringens, wala siya spores on ordinary media. Okay? So, it causes gas gangrene or myonecrosis, severe muscle destruction, and production of gas pockets in the tissues. It also causes food poisoning, post-abortion sepsis, intra-abdominal and pleuropulmonary infections, and enterocolitis. So, virulence factors of your Clostridium perfringens is the alpha toxin. It is a necrotizing toxin produced by all 5 strains of your Clostridium perfringens. We have your also lecithinase. The lecithinase will split lecithin to phosphorylcholine and diglyceride. So lecithin is an important constituent of the cell membrane. So kung wala siyang lecithin, it will cause cell destruction. Okay? Ayan. So strains of Clostridium perfringens, we have your 5 A, B, C, D, E. Okay, so the toxins produced by the following, no, are the si alpha kay alpha and theta, sa B alpha, beta, epsilon, type C alpha and beta, alpha and epsilon for D, and alpha and iota for E. So please familiarize lang this toxins, no, produced by the different strains of your Clostridium perfringens. Okay, so your clinical manifestation or clinical infection, wound and soft tissue infection caused by type A strains of Clostridium perfringens due to the alpha toxin. Ayan. Necrotizing enteritis, pig bell, or dumb braid is the ingestion of inadequately cooked pork. Ayan. The signs and symptoms is due to beta toxin of the type C strains. It also causes food poisoning, no, type A strains, with spores and minimal amount of theta toxin. So, laboratory diagnosis, again, specimens. Ayan, cultural anaerobic BAP. Clostridium perfringens sa BAP, guys, it causes double zone of hemolytic pattern. Ayan. Chopped meat glucose, abundant growth, positive o gas. Kani siya, guys, mura siya target, mura siya double zone of hemolytic pattern. Outside beta, inside kay alpha hemolysis. Alpha hemolysis, outside kay hemolytic, beta hemolytic. Ayan, target zone. Double zone of hemolytic pattern. They are saccharolytic. Okay, they ferment glucose, lactose, maltose, and fructose. We can also use egg yolk agar to demonstrate lecithinase activity. So, ayan, opaque yellow on the egg yolk agar. Lipase negative naman ang Clostridium perfringens, and they are non-motile. Ayan. So, this one, biochemically, ferment, glucose, lactose, and blah, blah, blah. Milk media, stormy fermentation, na ay crack, no? Kung makita ninyo, gas positive siya. Lecithinase naman, ayan, so naa siya'y opaque yellow surrounding a colony. That's positive for lecithinase. Okay, Nagler reaction is the biochemical test used to identify organism which liberate phospholipases. Ayan. So, Clostridium perfringens has phospholipase activity. It helps in the differentiation of Clostridium perfringens from other Clostridium species. Ayan. So, Nagler reaction. Okay, assignment pa ninyo. Okay, so half strict nimo og Clostridium perfringens and ang half kay strict nimo og type A antitoxin. Okay? Zone of precipitation or little or no precipitation. Ayan. O, reverse camp test. Okay. So, ganina, di ba, camp test positive to Listeria. Kay, the same man sila, pero lahi lang sila og known organism, si Rhodococcus equi lang. Tapos, kani, si Clostridium perfringens, reverse ang camp test. Nganong reverse camp test ang tawag? Kay, ang imo mang ipas, ang imo mang strict perpendicular sa imong unknown kay si group B strep, si Streptococcus agalactiae. Yan. So, arrowhead hemolysis, positive. Ayan. So, mo na siya principal again. Ayan. Is one. Reverse camp test. Okay. So, how do we, debridement of the necrotic tissue? So, it causes gas gangrene nga. Okay. Yan. So, aside from antitoxin therapy, we can also do hyperbaric oxygen to detoxify the wounds. Okay, next is, sumunod na kay perfringens, we have your Clostridium difficile. So, difficile is first isolated in 1975. It is antibiotic-associated pseudomembranous colitis. So, this is antibiotic-associated pseudomembranous colitis. Diba si diphtheria causes pseudomembrane on the throat. Si Clostridium difficile, it causes pseudomembranous colitis. Okay? Sa colon. Yan, colon perforations. Wait lang. It leads to sepsis and death. Yan. So, it is motile with perityphus flagella and non-encapsulated. So, the toxin produced by your Clostridium difficile are toxin A and toxin B. So, your toxin A is potent enterotoxin. Your toxin B is cytotoxin. Ayan. So, clinical infection, okay, antibiotic-associated pseudomembranous enterocolitis. The use of ampicillin, clindamycin, and fluoroquinolones will eventually lead to infections of your pseudomembranous, your Clostridium difficile. Ayan. Pseudomembranous colitis or ulcerative colitis. Ayan. Normally, ani siya, pag naana kay Clostridium difficile infection, pseudomembrane production. Okay. Ayan. So, specimen, liquid or semi-solid fecal specimens. Microscopy, gram-positive bacilli. So, culture media that we can use for isolation of Clostridium difficile is your CCFA or the cycloserine cefoxitin fructose agar. Yellow ang color niya sa colonies and horse manure ang odor niya. Ayan. Serological testing, we can demonstration of toxin. Ayan. Cell culture is the gold standard for the toxin identification. So, biochemically, mo niyahang mga positive reactions. Ayan. So, treatment is discontinue the administration of the offending drug. Okay, pwede po vancomycin and metronidazole and gastrointestinal stimulants. Stool softeners may be used, no? Kay, diba, obstruct na ang colon. Obstruction of colon because of the production of pseudomembrane sa colon. Okay, ayan. Miscellaneous Clostridia, ayan. Just read na sa book, no? Anaerobic gram-positive, non-spore-forming bacilli. Ayan. So, your Propionibacterium. So, your Propionibacterium, ayan. Or your, non-spore-forming na anaerobic. Okay, so normal flora lang sila usually. They rarely cause opportunistic disease. Genera under this one, Bifido, Liobacterium, Lactobacillus actinomyces, and Propionibacterium. So your Propionibacterium is a facultative anaerobic organism, non-spore forming and non-motile, irregular rods and branch forms. Optimum temperature at 30 degrees. Normal flora of the skin siya, GIT and ears, metabolize carbohydrate and lactate, and converts lactic acid to propionic acid and carbon dioxide. So your Propionibacterium acnes is the most frequent isolated bacteria in your blood culture. Okay? Yan. So, the Propionibacterium acnes. Normal flora of the skin, man good siya, guys. So, kung dili tarong ang inyong disinfection sa blood culture, okay, most likely ka na inyong ma-isolate. Okay, rare pathogen lang siya. Skin contaminant lang siya usually of blood culture. Okay, ko dapat tarungon gud ninyong disinfect inyong area. If makakos man siyang infection, no, it is associated with prosthetic devices causing bacteremia and endocarditis. Ayan. So, Propionibacterium acnes, opportunistic infection again, ayan, produces lipase that split the free fatty acid from skin lipids. So, your fatty acids causes tissue inflammation and acne formation. Ayan. So, mag-cause siya acne. So, on BAP, no, anaerobic BAP, yan, glistening, convex ang iyang culture and produces narrow zone of hemolysis. Ayan. So, this is your Propionibacterium in your gram stain. So, it causes acne fulminans and your acne vulgaris. Ayan. Acne lesions. Ayan. So, that's Propionibacterium. Okay. So, ano siya, guys? So, please read na lang on your Bifidobacterium. Ano to? Bifidobacterium. Lactobacillus, na-discuss na nato. Si Actinomyces, na-discuss na po nato. So, just read on Bifido and Eubacterium. Okay? Ayan. So, that's it for your gram-positive bacilli. Okay? So, let's end. Ayan. So, please study, 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 study. Okay? I will stop share na. Ayan, so manatag-discuss sa gram-positive bacilli. So I hope you will read your book. Okay, so supplement na lang ning lecture video, guys. Okay, and then please have submit your assignments no next week. Ay, delete no assignments man daw. So just make your assignment, no submission. Himua lang siya as your study guide. Okay, study guide lang to ninyo siya. Okay, so that's it for gram-positive bacilli. So I hope you have learned something and we'll have your discussion of Mycobacterium pagbalik na nato after the holidays. Okay, so yun, that's it for today and see you next week.